Welcome to the Glucose Neverlies podcast. I'm John Pemberton, a diabetes educator and researcher living with type 1 diabetes since 2008. I started the Glucose Neverlies website when my son tested positive for type 1 diabetes antibodies. In that moment, I realised I needed to build a resource strong enough to guide anyone who might care for him, and something I wish I'd had when I was first diagnosed. At first, that meant writing clear, actionable management guides, but listening to Peter Attia's The Drive taught me something. The written word often misses important details and context. Peter's conversations provide depth across nutrition, exercise, pharmacology, sleep, and much more. But I always find myself wondering, how does this information apply to people with type 1 diabetes? And this podcast is my attempt to fill that gap. Through my work as a specialist diabetes dietitian, I develop international guidelines, serve on global committees, publish in peer-reviewed journals, and present at international conferences. Therefore, I have access to the world's leading researchers and thought leaders in type 1 diabetes. That gives me a unique opportunity to combine lived experience, professional expertise, and cutting-edge science, and to share it all on here in conversation. That's why on this show you'll find the latest scientific evidence translated into practical steps, honest conversations on getting the most out of diabetes technology, insights on balancing nutrition, exercise, and sleep while living a full life, lived experiences on how to party, play sports, and much more, all in the context of type 1 diabetes. But an important note, this podcast is for informational purposes only. These conversations share knowledge and experiences. They do not create a therapeutic relationship. So, let's get started. Okay, so as you can see, Dr. Stefan Pleus has stepped in now for Dr. Guido Freckmann as we continue to part 2. So part 1 hopefully has given you a clear understanding of the landscape. And I think we discussed CE marking, unfortunately, is not a quality standard. But that doesn't mean if you only have CE marking the device, we don't know the risk. There are devices out there that have followed good study design procedures and have CE marking. And there are also devices out there that have followed study design procedures that don't allow us to understand the risk for insulin dosing. But the problem is with just a CE marking, you can't understand where they are. You have to look into the study design, which is not practical for the average person with diabetes or a healthcare professional. And we kind of then discussed iCGM is an extremely good quality standard, but certain aspects of the study design are not well defined. For example, are we suggesting to compare against venous glucose or capillary glucose? How many people with type 1 diabetes do you need in? What percentage of reading is needed here? So hopefully we've made the case of we need an international standard that defines both how you do a study and then also how you report the outcomes. And the amazing thing is, Dr. Stefan Pleus, who is now chair of the IFCC working group for CGM, is the perfect person to explain how those study designs should ideally look, and not just from this is what we think, from studies that have been performed, have been validated, have been assessed for robustness, which is very, very different to, for example, the FDA iCGM study design procedures were made up at the time based on what was currently available and what seemed directionally right, which isn't wrong, but they weren't based on prospective study designs to validate procedures that you're going to use in the future. They're two very different things. So I think I've captured the essence of that right. So Dr. Stefan Pleus, welcome. You also work with Guido Freckmann at the same Institute for Diabetes and Technology, and now currently the IFCC chair for the working group.
Yeah.
And obviously, unfortunately for you, you've been the sufferer of my emails at 6 o'clock in the morning, this long with bullet points, because I've got something in my head and I do have to give you a massive sense of gratitude because your tolerate is probably the right word, but very kindly point me out where maybe my thinking's off and also encourage me to learn a little bit more. So first of all, I want to say thank you for that. And really introducing, as we just described there, based on what we've just said, if someone wanted to think about the type of things that should be involved in a robust study design for a CGM evaluation, what type of things need to be in there?
Yeah, thanks for having me here. I'm very delighted to be here, and I actually appreciate your enthusiasm. Sometimes I take some time to reply, but it's really great to have someone as enthusiastic and motivated in the working group as you. Also with the direct connection to the people living with diabetes, especially we talked about that children who may experience unstable glucose concentrations more often than adult people with diabetes. Yeah. So the thing with the study procedures basically is the FDA, as you rightly pointed out in the first part, doesn't really specify how to conduct a CGM performance study, and the potential issue with that is that you can design your study to get good results, to avoid rapid glucose rates of change. And we know from experience and from studies that have been done over the years that many CGM systems perform worse at high rates of change than at stable glucose concentrations. And it's easy to understand why. CGM systems measure in the interstitial fluid, and comparator values are, as you pointed out, either from capillary blood sampling or venous blood sampling. And there's a physiologic difference that has to be taken into account. So if you have mainly stable glucose concentrations, then there's no need for a CGM system to have a good time lag compensation, for example. We have seen studies in the past where we had study participants provide glucose concentrations between 80 and 65 milligrams per deciliter. So that's, I think,
it's about 3.5 to 4, something like that. Yeah, something like that. Yep.
Yeah, 3.5 to 4.5, and that over a period of 5 to 6 hours. That is not really representative of what happens in daily life.
I mean, the way that I try and pick that into my analogy was, if you were going to pick a car and you wanted to know whether that car was safe for your child to be in with you, would you want a car that had only been tested in the slow lane at 40 miles an hour? Or would you also like it to be tested at 60 miles an hour in the middle lane and maybe even 100 miles an hour? Although that's over the speed limit, people go that speed. And not only that, that maybe they're weaving in and out of the lanes as well. Because if your car can perform well in those situations, you're covered. For all the eventualities. If you only know how it performs in the slow lane at 40, it may be really accurate and good up there, but you don't know. And do you want to take that risk when you're insulin dosing? Certainly for the children and people I support and myself personally, I would rather have a system where I know it's been tested like that than take a risk on one that hasn't.
Yeah, exactly. And that's a main motivation behind the work of the IFCC working group. We introduced a set of, we call it comparator data characteristics. So it's basically, if you do a CGM performance study, the comparator values you obtain have to fulfill a specific distribution, both regarding blood glucose concentrations, which has been established some time ago, which also the FDA looks on, but also a minimum amount of data at high rates of change, specifically to address impending hyperglycemia and impending hypoglycemia, so that all of these situations where you want your CGM system to be very accurate, that these are covered to a degree that is maybe over-representative of what the typical user observes in their typical day. But these are situations that happen in people with type 1 diabetes, and as you pointed out, especially in children with type 1 diabetes.
Yeah, I mean, the way that I would frame this and to think about is, and you're quite right, because people have said this to me before, and I tried to explain, like, yeah, but you're going into a study and asking them to have food without insulin, it goes up, and then give a little insulin, come down. People don't live a life like that. Well, of course they don't. But if there is a chance that it will happen, and unless you test a system like that, you're not going to know. So, and the idea of a standardized procedure, as you've already started to explain, is it's not what will represent in real life. It's to represent, when you put it under stress test conditions, how does it perform, and does it meet what we need it to meet for insulin dose decisions. So they're two very different things, real life and study design conditions. The study design conditions are very, very simply assess how accurate across the full glucose range and at fast rates of change, not how it will perform necessarily in real life, but cover all of those situations that potentially will happen in real life.
Yeah, exactly. If we know that a CGM system performs well under challenging conditions, then it will perform well in the average daily life situation. And just to add to that, the average daily life situation might be different from one person to the next. So who's to say that the study population that is enrolled in the study is really representative of all those cases of glycemic control that people with diabetes encounter?
So I think I'm right in saying, because I've sat in enough meetings to hear this enough times, is when I say the current guidelines, if you take the performance of continuous glucose monitoring metrics, is within your study, out of the 100% of readings, at least 8% need to be below 4.4 or 80 milligrams per deciliter, and at least 5% or more need to be above 16.7, which is 300 milligrams per deciliter. Okay, so that sounds good. However, if I was a manufacturer and I was clever, what I would do is I would bring them in a lab on one day. They would start with a glucose level of 4.5. I'd just give them enough insulin to drop down to below 4.4, sit them in there, collect all my readings, and then just up above. And the next time they come in, I would start them at 12 and I would push them to 17 and stay there for a little bit and then drop them back down. So you cover the readings, but you don't get the rising fast and falling fast, which means when you do your accuracy results, it will look extremely accurate. However, it's not, again, letting us understand the risk of what will happen when these fluctuations go quickly. So with the recognition of that, what is it that the IFCC are proposing for these percentage distribution ranges, which advance it from the current model?
You're completely right. We know that manufacturers at least tried that approach.
Why wouldn't you though? Because if they're the rules, they're the rules. And I was a manufacturer and I want to get the most accurate device. It's within the rules. So it's kind of like, for me, it's like, don't complain about the manufacturers, change the rules.
Exactly, yeah. I completely agree. So within the IFCC working group proposal, what we say is you need to have a certain number of data points, at least 7.5% below 70 milligrams per deciliter, that's 3.9, and above 300, that's 16.7 millimoles per liter. And you also need to have 7.5% of data points each in what we call the alert low and high regions. It's not the most
sexy terminology.
No, but we selected them because that's when you want your CGM system to accurately alert you to an impending hypo or hypoglycemia.
Okay, so you got 7.5% when you're low, 7% when you're high, 7.5% when you're rising fast towards high level, and 7.5% when you're dropping fast towards hypo. And then the rest can be in the middle?
Yes, the rest can be in the middle, and that's basically 70%, 60 to 70% of the data will be found somewhere in the middle. There may be rapid rates of change, but at non-critical glucose concentrations, for example, dropping rapidly from 16.7 millimoles per liter is not as risky as dropping rapidly from 5.5.
And so just harping back to the first conversation I had with Guido, and we mentioned the study they performed against the market leaders A, B, and C. And within that study, we described on the same study day, rather than what we just described, you have to make them go high with a meal and then down low, because that's the only way you're going to obtain these 7.5s. You're not going to attain these 7.5% if you bring them in and just go high one day and low the next. And that's the key, isn't it, is to standardize the study design. The in-clinic study days for 7 hours need to be go high first and drop down on the same day to get the distribution correct.
Yeah, so other approaches are possible. We know that, we know that from the feedback we got. What we found the most easiest approach is give our study participants a carbohydrate-rich meal with fairly rapidly resorbed or absorbed.
60 grams ish, right?
No, it depends on the daily energy requirement.
Okay.
So 25% of the daily energy requirement, and then 65% carbohydrates, 15% protein, 20% fat. Yeah, and then our participants they eat that meal, and once they go beyond 250 milligrams per deciliter, then we apply the insulin bolus that they would normally have applied at the beginning of the meal. We don't increase the bolus because that might increase the risk of hypoglycemia. We don't need it as also, so they go up at 250. We dose until the insulin action is at its peak. We often have surpassed the 300 milligrams per deciliter, 16.7, and then they naturally come down quite rapidly, not as rapidly as they went up, but quite rapidly on the way down. And any glucose manipulation procedure, even the slow ones, need medical oversight. So you need to have a trained physician on staff at the study site. We do that obviously as well. And our physicians, they look on the glucose values every 15 minutes. We try to once we are nearing the 70 milligrams per deciliter, we already give small amounts of carbohydrates so that to minimize the risk of very low glucose concentration.
Sure. Yeah. So obviously this is going to take a bit of practice and skill. And one thing I think I've picked up from the manufacturers is obviously they use multiple sites. Maybe they use 10 different sites and they have different skill levels. And one of the concerns, quite rightly, is how if this was to be embedded tomorrow, which is obviously not going to happen tomorrow, but it would take some training and some time. But as I understand it, the procedure's been validated. They've just the discussion there, because I remember reading the percentages have been laid out. Even like tools have been developed for the analysis. So everything's been done. And I think I understand the manufacturers saying, well, there's going to be pushback from the sites because they're going to say this is difficult to do, which is completely understandable. I'm just thinking from a person with diabetes. So it was kind of like, oh, people will never be able to do carb counting, or they'll never be able to do insulin dosing, or 4 or 5 times. They won't be able to work out correction dosing. When they're using CGM, they won't know what to do with the arrows. It's too much information. Well, actually, you adapt. You adapt to what is best. And of course, at the beginning, there will be some challenges with that. But if the idea is to move forward to aiming at the same place, whether it's capillary or venous, ensuring that study endpoints can actually be trusted for future therapies, and also ensuring that people with diabetes have consistency when they go back to their clinician in terms of therapy adjustments and the clinicians feeling confident, it sort of feels like it's a necessary thing to undergo that difficult change, because if we don't, we're going to continue to have these challenges with CGMs coming to market that we don't know the risk. We're going to have people with diabetes who are sometimes using a venous aligned CGM and then sometimes using one and getting frustrated. Differences between finger pricks and CGM. It's all very messy and very challenging, and it can be managed, and it will be a bit of challenge to adopt those. But I do see it as a fair standpoint against, but at the same time, change is possible. And if it's worthwhile, and I think we're kind of building to the case where we're at a point where it's not worthwhile, it's becoming almost essential, in my opinion. That's my opinion.
Coming back to the first part with Guido, what we have seen time and again is that different CGM systems provide substantial, or can provide substantially different CGM readings. And that means that the quality of diabetes therapy that you as a user of CGM systems receive depends on the system you use. And that's not a good idea. And our main issue with the FDA iCGM special controls is that this is not addressed there. So in our opinion, at least Guido's and mine, and also it's a majority opinion in the working group, we need to move away or beyond FDA iCGM special controls. We cannot rely on those alone because we know that they are insufficient in addressing these discrepancies.
And I think one point important here is to really think about science. For me, this is just my opinion, is 2018, it was cutting edge, absolutely brilliant, and it really helped revolutionize the field.
Yeah, it did.
We're now in 2025, the science has changed. There's new science out which shows capillary would be the better option. And we're going to get into a minute about how we measure that. But we're not saying, I don't think anyone is saying, that the iCGM is not a very good quality standard. It absolutely is. What we're saying is, now that we understand more, we're using more systems and there's more challenges coming. It's time to go beyond that and move towards something which is global, robust, systematic, and reproducible, so that everyone gets the benefits that we're thinking about. And with that is a good point to jump into one of the iCGM things, which is it needs to be a lab grade analyzer for the comparator. So back in 2018, I think everyone would agree with that. But tell us about the work that's been done with capillary glucose testing and the bias, how that is, and how that can be reduced in a systematic way to ensure that capillary glucose is certainly equivalent, if not superior, to venous glucose measurements.
Okay, so you're right there. The science has moved on, and we now know more things about how to address this topic properly. For the venous versus capillary topic, you already talked about with Guido about that. And I think there can be made a fair point towards capillary if there is a consensus that we should all move towards venous. I'm fine with that. At least it has to be the same for every CGM manufacturer to avoid these discrepancies. We can talk about all that.
Yeah, I
happens
mean,
afterwards.
Yeah, I guess kind of, and I would agree with you there. My preference, not that I'm the czar of continuous glucose monitoring, would be capillary because, as I said, as a user, you're told in that information for use, if you feel different to CGM, do your finger prick. So that's your gold standard. That's your most accurate bit. So you want it aligning with that, otherwise you have problems. It's going to tell you your exposure to post-meal glucose better, which is going to give you a complication risk and allows you to drive at a target better. So for me, and also the trials thing is kind of, it's irrelevant. The trials will be fine as long as it's to venous or capillary. But from a user perspective, capillary makes sense. However, if that can't be managed and we do go to venous, one thing that will have to happen is 70% timing range will not be the equivalent anymore, because it'll have to push 75, 78% timing range to get the same complication future, which is fine. But if I said to people with diabetes now, all of a sudden, when we standardize, you have to push up to 78% timing range to get the same benefit. It's hard enough to get 70% in range.
But it will be easier.
Yeah, it will be easier, potentially easier.
But that's a conversation that has to be held when that is decided.
The main thing that I understand for the challenge for why not capillary. So there's two things. One is that the FDA doesn't recognize capillary glucose from point of care or finger prick monitors, because the perception is that the level of bias and inaccuracy is too high to be a comparator. So that's one thing that you can unpack. And then the second one alongside of that is the practical implications of doing so many finger pricks on an individual during the study. So I think they're things that need addressing. So tell me about what your current feeling is around those.
Just one addition. The FDA, at least that's my understanding. It's not published anywhere, but it's my understanding that the FDA thinks that, or is of the opinion that venous glucose concentrations better reflect the physiologic, let's say, needs than the capillary ones. That was also this discussion we had with BG meter manufacturers some years ago, who got feedback from the FDA on that topic, and that is also something to unpack. I think capillary samples capillary sampling can be done in these clinical performance studies, and the thing is, within the working group, one of the main goals is that we think it's important to align CGM readings between different systems, and we also know that not only is there this systematic difference between venous and capillary concentrations, but also there can be systematic differences between different brands of glucose meter or laboratory analyzer, and there can even be systematic differences within the same brand. Everybody who's old enough to regularly finger prick, do finger prick testing knows that from one test strip lot to the next, there can be a systematic bias of a few percent. But the same is true for laboratory analyzers. Depending on the reagent lots and the calibration of the lab analyzer, there can be differences. And that's why we from the working group think that it's important to assess this bias within the context of a CGM performance study, because that bias is not set in stone. It's not consistent over several years. You have to assess it within the context of your CGM performance study and then correct for the bias using reference materials. In the official wording is higher order or reference materials or methods of higher metrological order. That's too technical for many.
Okay, let me try and unpack it into terms of that I understand it. So what you're saying, as if I understand it, is the belief that, for example, a lab grade analyzer such as a Yellow Spring instrument or a Cobras Integra is that alone from different ones is accurate enough to do a performance assessment. Now, if I'm right in saying my statistics, not that I really understand it, but you would want a bias less than 2.4% when assessing glucose measurements between things. Is that right?
Yeah, so we want to have a bias of less than 2.1% in comparison to higher order reference materials.
And that would methods. Be what would be this highest order material? Would that be like magnetic resonance glucose?
It's mass spectrometry. That's an accepted reference method, and there's materials like the NIST standard reference material, glucose in serum, which are accepted as higher order reference materials.
Okay, so we're getting totally into the weeds, but just if I can bring this into my head, what you're going to do is you're going to have these Yellow Spring instruments, let's just say in a venous one. You take that, and then what you want to do to assess the bias is see how off it is to this higher order reference material between the different systems. And essentially to do that, you've got these higher order materials that are like exquisitely produced to be exactly the right thing, and you do that obviously within the lab. So you've basically got a few levels to ensure, and then you can correct for that, can you? If there's a difference, you can apply mathematical corrections to bring all the readings back in line. And so that's obviously beyond my mathematical things, but essentially, as I understand it, it's like a calibration. It's basically a calibration to make sure that the comparator that you're comparing your CGM against is actually measuring as close to the glucose in the body as possible, and you need to do a calibration for that. And without that, current Yellow Spring instruments and Cobras Integras don't actually meet less than that 2.1%. So even in the FDA studies, potentially that bias is being breached.
Potentially, yes. So
not certainty, but very potentially from the research you've done.
So what this bias correction intends to do is to correct the comparator results as close as possible to what you would call the true glucose concentration in the blood. Every measurement method has inherent error, so we don't know what it is, but that's the best representation of the true glucose concentration. And we want to align with that across studies, across manufacturers, because we think that once a CGM system should look good, its performance should look good in comparison to the comparator method. And if all comparator methods show the same level of trueness, trustworthiness of glucose concentrations, then we will also see an alignment in the CGM readings. That's the thinking behind that.
And right in saying, so we just talked about the lab grade analyzers, you can do that, but you can also do that with the readings from the point of care,
Yes.
capillary, and
Yes.
blood glucose. So
Yeah.
obviously, if you can do that from those, then that argument from, for example, the FDA that it needs to be a lab grade analyzer for accuracy actually doesn't stand, because at present we know that the bias, even from those lab analyzers, is above that. So you're still going to have to do the retrospective correction anyway to get below that.
Exactly.
So why not go to the next step and use capillary, because you're going to have to do something anyway. Why don't you just change it and allow this?
Yeah, that's a good point. To coming back to that, we have a few lab analyzers at the Institute, and we because we use them also as a comparator in BG system accuracy studies. We often contact their customer service and say, well, we see that there's systematic differences here and there. And when we tell them that we aim at below 5% bias, they say, well, that's not a usual case scenario. We are not prepared for that level of performance to be consistent across all that's manufactured by these lab analyzer manufacturers. And I understand that, at least in Germany, for example, the official guideline for a lab analyzer to be sufficiently free of bias, you have to do an external quality assessment scheme at least 4 times a year. And the goal there is plus minus 15% from the reference method value. And we are now talking about plus minus 2.1%. So we cannot expect the lab analyzers to be good enough. And that's also why I think that what the FDA is asking is insufficient in aligning CGM system readings.
So I guess there's a lot of technical information there, but bringing it back from as I understand it, is at present, even with the lab grade analyzers from the iCGM, you're going to be breaching that 2.1% bias, which means, and the reason why this is really important, and you can obviously do that with capillary as well, so there should actually be no argument if you do it correctly and apply the retrospective correction to capillary versus venous. If you could choose what you want, if it was accepted. But the reason why it really matters, I remember reading the paper that you were obviously involved in, is if you look at the mean absolute relative differences of performances before correction and then after correction, there's a 2% difference in MARD. That's, when you think about the public perception and the marketing from something to go from 10% to 8% or 9% to 11% from a MARD, from a user, a healthcare professional, if something all of a sudden went from 9 to 11, you'd be like, wow, that's terrible. Or if it went from 11 to 9, you'd be like, oh, that's improved. And that's why the standardization is so important, because if you know the bias is less than 2.1%, then you don't have this discrepancies of MARD based on whether the analyzer was reading accurate or not.
Yeah, exactly. These discrepancies will still be there. No analyzer is perfect, and no bias correction is perfect. But we expect these differences in, be it MARD or the agreement rates percentages within specific deviations, these discrepancies will be much lower, provided that the CGM systems actually provide consistent results.
Okay, so let's climb out from deep in the weeds back up to the top to where people have an understanding about right. So I'm going to try to summarize. In an ideal world, a future study would have at least 75, maybe 80% of people with type 1 diabetes. Somewhere around 100 people, there or thereabouts, would be adequate to get the distributions and make sure that it's statistically effective. You need the meal and insulin challenges on the same day as we described to get these 7.5% readings here and there. Either venous or capillary, but retrospectively calibrated to get the bias below 2.1%. Obviously, in my view, I think that most people's view, the capillary would be ideal, but that's up for discussion. If you have that study, then what you're going to have is a set of results to then report how accurate these things are. And I think a really important point to drill home here before we get there is we've been seeing MARD, how different on average is a sensor reading to the true comparator reading. They've gone from 15%, 12%, 10%. We're now down at sub 8%. If we applied this robust methodology with faster moving glucose levels, basically the sensors are going to be looking less accurate than the previous research. Nothing will have changed other than the study design procedures will have tested the sensor robustly.
Yeah, exactly.
So if that study came out tomorrow, the risk would be, from a company perspective, and I totally get this, we used to have a MAD of 7.8% and now it's gone to 10. I can't market that. So presumably there needs to be a new set of metrics that moves away from the old ones, because we're now looking at how these systems perform when rising high, when going low, because that's what we're bothered about. We're measuring them. So if we had a complete different set of metrics, we can say, these metrics belonged, the old ones, to the old study designs. Very variable. They don't really tell us a great deal necessarily about the consistency. If we have robust study design procedures, and now we have a new set of metrics to match these study design procedures, we'll know what's good enough. And then in the future, presumably, if you meet whatever it is, it's an ISO standard stamp. And then the people who are looking after people with diabetes and the people they don't need to know all this stuff. They could just go, it's got an ISO. We're happy with it. But so tell us about how the metrics might look different to what's currently there. I know they're still under discussion, but how would they look different?
Okay, so we need to move. So we know, again, from experience and from literature review, that the level of CGM performance in a specific study depends on the study procedures and the study design. So we cannot expect that for example, FDA iCGM special controls or the magic MARD of below 10 will fit with the new study design, because we know that even current iCGM systems, we know they are safe, we know that they work well. But even those with a challenging study design will have trouble achieving iCGM special controls or MARD below 10.
And that was shown, wasn't it? In that study, the same set of studies that we talked about before, where you did the robust study design, when you actually pitched them against iCGM, granted the number of people was only 24, therefore the variability may change. But in reality, it's unlikely that it would have hit every single metric. Maybe hit a lot of them. So and this is a really important point, is that obviously the iCGM, people who've got iCGM, the manufacturers, don't want to let that go because it's a quality standard.
Yeah, exactly.
But it's a quality standard built on good theory back in 2018. But when the science has moved, it's now not based on robust study design for consistency. So and there's nothing wrong with that. So with a new study design comes a new set of metrics, and we can say that was great from 2018 to 2030, but 2030 onwards, the science has moved on in 12 years. Now we need these set of performance metrics, and it's okay that they don't meet the old ones because the study design is different.
Yeah, exactly. So just to make that point clear, we know that the systems we analyzed in our study last year, and where we showed that not all of the iCGM criteria will be met with our study design by these systems, we still know they are good systems. And so and that is something that's very hard to convey to people who work with people with diabetes. You have to look at the study design to make sense of your performance parameters. And that's why we need a standard, because with a standardized study design, we know that these performance metrics become much more comparable from one study to the next.
And I think this is the important one, because I spend a lot of my time, because I'm a total nerd. When we come to these conferences, we're obviously EASD. I'll go around and I'll speak to the different manufacturers, but speak to different people within there. Now, if I speak to the medics and the data team, they get this and they understand it. If I speak to the marketing and sales team, they're like, you're going to take my MARD from 7.8% up to 10%. Hell no, that's not happening. Like, and you get it, because they're not meant to understand that. That's what they're not skilled at that. But if we said to them, your system meets a robust ISO standard, and that's all you need to know, and you don't need to know the rest, it makes it easier, because then once you achieve that, then we can get to actually marketing, not on massaged accuracy statistics, actually what the system does. What alerts does it have? what new features does it have? Because we know it's good for insulin dosing. We want to know what data you have on how it works for people. We don't want to be marketing on, oh, we've now got a MARD of 5% because we made people sit in the lab and do nothing for 7 hours, and we've got a really accurate MARD because we've managed to do a study design that meets the current criteria. And if I was a manufacturer, why would you currently do a really robust study design? Because you're going to come out with a MARD of 12% and your sensors are going to look terrible. So you're not going to do it unless it's mandated.
Yeah.
And that's the bottom line, isn't it?
Yeah, exactly.
So I think we kind of got to a point from the 2 episodes where we've explained the CE marking situation. We've probably, hopefully defined that iCGM in 2018 was the best in class and we're happy with it. We're now in 2025. We realize the science has moved on and it's time to implement the new science, looking at an ISO standard, maybe capillary, maybe venous. If it goes to venous, maybe we need to revisit timing range metrics. If we stay with capillary, obviously there needs to be some alignment with the FDA to understand now the necessity for a lab grade analyzer can actually be overcome with the new science. If they're not willing to do that, because things are set, then maybe it is venous glucose, but with the new bias corrections, study design procedures, and maybe they need to revisit the performance metrics. Obviously, I'm pretending like I'm in the FDA here. But either one of those 2 things needs to happen so that then an international standard can be put in place. So at some point in the future, when a manufacturer comes to see me at Birmingham Children's, they have an ISO standard for insulin dosing for children. I don't need to know all this stuff. I just know it's been done properly. What do you see the potential timeline? What do you see as the biggest barriers? And what do you think might be needed to overcome those barriers? That's a big question. But I think it's important because I think we've identified there needs to be change. But the roadblocks and the pathway to there is not an easy one, but in my opinion, it's a very important one, and a lot of things have to happen to get there. So in your view as chair of the IFCC working group, what would you like to see? What would you see as the barriers? And maybe we can come up with I can come up with some solutions.
So regarding the timelines, an ISO standard takes a few years to be established. We have been in talks with people at ISO about initiating that work, and that already has taken some time. But usually an ISO standard takes about 3 to 5 years from formation of the project team until it's established. And then once it's published by the ISO, it's not necessarily introduced in the regulatory frameworks in different jurisdictions. That also has to be done. So I think when we are talking about an ISO standard, we are talking about likely 2030.
Okay. So that's if it runs smoothly.
Yes.
So what are the potential roadblocks along the way? What are the big ones? There'll be not every single one, but there may be the 2 big ones.
So a very big roadblock is actually the FDA, because most CGM manufacturers want to sell their CGM systems worldwide. That includes the United States. If the FDA cannot be convinced of the need to adapt the current regulatory framework, that would either lead to a situation where our proposal is rejected completely, which I don't like, or the alternative would be for manufacturers to perform different studies for a US market and a European market. And I can understand that manufacturers are not interested in that. These studies are expensive. Maybe they're more expensive in the United States than here in Europe, at least from what I heard recently from these manufacturers. But it's still a financial burden to them.
Sure.
And I can understand that they hesitate to adopt a new guideline. So that's the big one. That's the big one, yeah.
So let me just unpack that a little bit. And I think actually for the listeners, both healthcare professionals, and no doubt manufacturers will be listening to this, potentially people from key organizations. If what you've heard makes you compelled to think standardization is really needed, and one of the major challenges is obviously alignment between a US versus outside of US, then obviously for an iCGM, and it would need to be a regulatory change. And the question is it important enough? Well, for me, if the future of clinical endpoints for glucose-based studies is pushing towards CGM, that alone is absolutely massive. Forget about people with diabetes and their experience. We're talking about the future therapies that are going to determine people's health will be based off CGM readings, some aligned to capillary, some aligned to venous, no consistency, trial results not being replicated, messes with science, wasted of millions of pounds for pharmaceutical companies. That alone should be a financial incentive. You hear at the ESD, it's GLP-1 this, SLG2 this. If we're going to move towards time in range, average glucose, whatever, and meta-analysis are performed on those, if I was reviewing, I'd be like, no one can trust that result because I know you can't trust that result.
That's a good point. We at the Institute do a lot of peer review for publications. That's also something that we encounter. We had a study to review. I'm not mentioning any details because
it's not about calling companies out. It's just about these are the problems.
Yeah, the main problem was that the control group and the intervention group use different CGM systems, and that alone makes, to put it blunt, that makes a study worthless.
It's a rejection. Yeah, it's a straight up rejection.
Yeah. And that's
someone's PhD life work has been spoiled.
Yeah, exactly.
What could have been a great piece of science, and because they don't know this stuff, they're not necessarily going to know that. Whereas if it was ISO standardized, that'd be fine.
Yeah, exactly. I'm not suggesting that the people weren't intelligent enough to do that study properly. It's just not advertised by the manufacturers that there are these differences. And maybe we from the working group have to do a better job in raising awareness.
That's why we're here.
That's why we're here. Exactly. Yeah. And so these are important topics. And then once we talk about using CGM in diabetes diagnostics, there has been a recent publication about the use of CGM when diagnosing the progression of type 1 diabetes. That's all fair, but with the current regulatory frameworks, including the FDA, that's not possible, in my opinion. We need to do better.
Well, yeah, 100%. I mean, I said on the previous episode of Guido, my son tested positive for type 1 antibodies. He would be one of those people potentially in the future that you are using CGM on to determine when insulin doses and progression is going. So based off the study that yourself was involved with, the Guido, and those market leaders, if he was using the market leader that was aligned to venous glucose, you would think he's never having diabetes because he'd be reading time in range, time in tight range, and you'd be too late on therapy. Whereas if you're using one of the other ones, you'd act quickly enough. That's not good enough, like, for the family. So if you're someone with diabetes, number one, that means you're going back to your clinician and you've potentially got a time in range that doesn't represent your complications risk compared to another. If you've got a young person who's tested positive for type 1 diabetes antibodies, and there's going to be a lot more of those people because these screening programs are going out all over the world and there's going to be large numbers. If they're going on different devices, they're going to think they're either progressing or they're fine. Yeah. So that's the second element. There's the trials, there's the diagnostics, and then there's the people like me who already have diabetes who are getting, maybe changing brands of CGM because their AID system that they're going to use uses a different CGM, and all of a sudden they're going to be hitting the same timing ranges, but the HbA1c is going to be different and the complication risk is going to be different. So I think the FDA roadblock, if you're a person with, if you're a clinician supporting people with, if you're an organization that is a patient-driven organization, it's something you really need to be thinking about jumping up and down about, because the manufacturers who are currently market leaders have got no incentive realistically to challenge the FDA, because why would you upset the apple cart? However, if the people who they support, the clinicians that they support, and the organizations that they sell their wares to went, hang on a minute, this doesn't sound quite right. Let's put some pressure on for you to help the manufacturers on the FDA, because there is a need to change. That's how change is going to happen. Change isn't going to happen by a small working group in the CGM banging on about the science, and we're validating. It's going to happen by people who really think this is important, understand why it's important, and be willing to get some skin in the game and put some pressure on the FDA or the regulatory bodies in their local country. I've been in contact with the MHRA a number of times saying, look, this is the situation.
Yeah.
It is not good. It's dangerous potentially, and the future is going CGM. Let's get standardization so the future looks happier than a potential of drugs being approved or not approved based on readings which are not comparable.
Yeah.
And that, again, is where we're heading, unless something goes in place. We're not there yet, and there is a big chance to make it by 2030 very different, but it's going to take more than just a small working group and a few people. It's going to take a robust effort to support the manufacturers to get some weight behind it.
Yeah, that's right. And just to reiterate that point, what the FDA did in 2018 was great for the time, but the limitation is it's for integrated CGM systems. So basically, or the most common scenario is AID systems, insulin dosing. So insulin dosing works well with iCGM, and I don't doubt that. And that will be, the iCGM will work with insulin dosing for the next century, maybe.
Yeah.
But we are talking about progression in the science and in the application of CGM systems. And when FDA issued the iCGM special controls, I'm pretty certain they did not think of CGM use in the diagnostics of diabetes.
It wasn't a thing then.
Yeah,
it wasn't a thing. The science has moved.
Yeah.
Testing needs to be improved.
Yeah.
So one last point that we'll cover before we close off, because I think what we've led up to is actually going to answer this question easily. But we're obviously EASD. And there is the iCGM, and now there's a suggestion for the eCGM, which is essentially adopting the iCGM, not completely, with a few caveats, but extremely similar, both in terms of study design and performance metrics, into Europe, so that then we're, quote unquote, got systems that are comparable. So obviously the downfalls of that, I'll mention ones that I think we've talked about, and you can tell me wrong. We've got all the study design problems and inconsistency. We've got the fact that some are pointing at Venus and some are pointing at there. So we've got all that stuff. But then there's a second and even more important point for me as a user is, if you brought that in tomorrow, we would only have 3 different types of devices available, or maybe 4. And also it would mean for a company to do a level of a study design and take it to there would be cost prohibitive, which would mean innovation would go no competition, monopoly of the market. That is not healthy. That is not healthy for me, who wants the development of CGM to really kick off. And so to just kind of transport that into there sounds like a good idea, because it would have short-term robustness. However, for example, the Rush Smart Guide, the Care Sense, which at the moment have done fairly robust study designs, would be kicked out. Well, they've actually done a robust study design, and I personally feel happy with the risk that those present. Now, there's some others with CE marking that do not, and I can answer those with questions, but just shoehorning that in and leaving it there is going to leave us in the same problem, apart from we're going to end up with less people coming to market, more difficulty in coming to market, and less clarity. And also the other thing in the EU, as far as I understand it, is the only way that you will get notified bodies to follow a study design protocol is if it's an ISO standard, not if it's a clinician consensus.
With an ISO standard that's harmonized with the medical device regulation, that's your best bet. Yes.
Yeah. So, I mean, it's just that I think it's so important that, as we said, iCGM 2018 was absolutely great, but do we want to bring a 2018 thing here and plonk it in 2025 when we already know the science is saying different? We just need to get on with cracking on. Maybe it's an okay interim solution, but you still then end up with ruling 2 or 3 good devices out and monopolizing the market in the meantime for 2030. It's not an easy thing to solve.
Yeah, and especially so you have to differentiate between allowing any iCGM device on the market in Europe. That's the one thing. And the other thing is notified bodies having to look for iCGM compliance, because I'm pretty sure that the notified bodies here in Europe will not provide the same level of consistent review that the FDA does.
I mean, just as I described to Guido before, if you're a general practitioner in your local care center and you go in, you've got 40 different things to look after. So if someone comes in with a very specific thing like type 1 diabetes, there's no way that person is going to be able to look after you properly. If you go into the hospital tertiary center where you've got specialists in there, you're going to get the care. So again, they're not going to be able to do that. Whereas if you have ISO standard study designs done by approved sites, then they just take that to the CE marking and go, yeah, I've got the ISO, I'm good for non-adjunctive therapy. So obviously I've got the rose-tinted glasses on and get excited, but I don't know, I feel like over these two things, the way that I would summarize it, and you can help if I've done this justice, is we're in a situation where CE marking can't be trusted on its own as a quality standard. You have to look into devices with CE marking only to determine whether they've got a quality thing. And I will actually put a plug here for the chat that I helped the Diabetes Specialist Nurse Forum done. A simple 5 questions, some of the things we decided, discussed about, meal insulin challenges, 75% people with type 1, etc., etc. And it scores each of the current available manufacturers on a score of 1 to 5. If it's got a score of 4 or above, this is, for now, a robust enough study design, and you're happy with the accuracy metrics, go and have a look. Anything less than that, you're not going to go the risk. But that's some way to help you navigate the current market. iCGM 2018 was absolutely brilliant. It's still valid now and will continue to be valid, but has study design problems. The future to make sure that CGM can be used in primary endpoints for clinical trials, can be used for diagnostics for people developing type 1 diabetes moving forward, allow people with type 1 diabetes to ensure they understand what their complications risk is. The only way for that is standardized study design, ISO standard to implement across the board. But the big stumbling block that we're going to hit is the alignment of that with the FDA, which is going to require a concerted effort from people with diabetes, healthcare professionals who support people with diabetes, patient-centered organizations who believe this is really important, big conference organizers who recognize this is essential for the future of diabetes care moving forward, to go, let's support the manufacturers who would like to make a regulatory change in America or some negotiation, at least have a conversation, get some alignment, and then we've got a chance. Is that a fair summary?
Yes, I think so.
So hopefully you found that helpful. That is going to be a massive deep dive and something that you may have to delve into, but hopefully that summary at the end just pulls everything together. And if you believe what you've heard is reasonable, and you should always have a critical eye, of course, we'll put things in the show notes, links to the key papers, the 3 studies we talked about, links to all the documents, so you can make your own independent assessment, because you may believe that we're biased. We are biased. We sit on the IFCC, we're a working group. We believe standardization is the correct way forward. You should absolutely have a critical eye. But I personally believe you go look into that further, you will find we won't be too far away from the truth, or as close to the truth as possible. And it's worth having your own assessment. But if you believe it's important, the future of diabetes care needs your help, essentially, in terms of thinking about how to navigate this moving forward. So hope you found that an enjoyable watch. And certainly I want to say thank you to you for describing that in a clear way, and also for the ongoing emails you're going to have to get from me and decode. So thank you very much for your time.
Thank you for inviting me.